Hello again, Dr. Mashburn, and so we're going to continue learning about the blood by talking about the erythrocytes. All right, so the erythrocytes are your red blood cells, okay? So we haven't had lab yet, but just to get you thinking about what these look like as we're working toward the lab portion in a few lectures. This is a smear of a blood slide, and remember, blood is cells inside of a liquid matrix. So the erythrocytes are all these little pink discs here, okay? The big discs, these are your white blood cells, and we're going to learn how to identify all of those later on. But right now, just the red blood cells are these pink discs. And these pink discs are what are called kind of like these, they're like donuts or biconcave discs. And their shape, remember, form follows function. They're very good at what they do. Because they have this particular shape, they're able to bend and twist and flex. So if you remember, earlier I talked about how blood goes to the heart in a vein, then it goes to the heart and then out to an artery. And then the arteries are going to connect to the veins and capillaries. And you'll learn more about this in later on our couple of lectures, in a few more lectures. But these capillaries are really small. They're like the diameter of one cell. And so by having this particular shape, they can bend and twist and go through these capillaries. This biconcave shape also gives them a very large surface area relative to volume, which means they can hold more than they could if they were in the round shape, the perfectly globular shape. These cells are a nucleate, which means there is no nucleus. As the cell matures before it enters into the bloodstream, it spits out the nucleus and all the other organelles. That gives it more room, more surface area, more surface volume, or volume in the cell to carry oxygen. So more hemoglobin, which allows it to carry more oxygen. If you look at a red blood cell, most of what the cell is made up of is made up of hemoglobin, which is a protein that has an iron attached to it, which allows you to carry oxygen. So 97% hemoglobin with a little bit of water that's filling up that cell. There's no mitochondria, so there's no ATP being made. And that's really important to know, because you're thinking, oh, red blood cells, they carry oxygen. I bet they make a lot of ATP. Well, they don't. They only take oxygen to cells that need it. They don't use the oxygen themselves, so no ATP is needed or consumed or made by the red blood cells. Again, they're the size, their diameter is such that they can squeeze through capillaries. They're filled with hemoglobin, which is abbreviated HB for gas transport. The hemoglobin allows the cells to carry oxygen and carbon dioxide. These plasma membranes of these cells have something called a spectrum, and the spectrum is kind of a bendy protein, allows the cells to be flexible so they can squeeze through tight places. And the red blood cells are the major factor in blood viscosity, or if you want to say thickness. So if you had just too many red blood cells, your blood can actually become kind of sludgy. So it gives you the viscosity of your blood. Think about it's kind of the thickness of the blood is due to its viscosity. And so again, the red blood cell is just a great example of that form follows function. So the form that it has kind of tells you what the function. It's just a bag of hemoglobin to carry oxygen, basically. Again, the main function is gas transport, okay? And it does that with the help of hemoglobin, which binds to oxygen. But it's not tightly bound. It's reversible, so that it can bring oxygen, it can carry the oxygen from the lungs through the body and then release it to the tissues. Okay, so it is a reversible reaction where you can bind oxygen and then release oxygen, which is really good. If you think back to chemistry, you wouldn't want a irreversible chemical reaction where you bound the oxygen, took it to the tissues, and then it didn't come off. So you have this really nice reversibility with oxygen binding. Hemoglobin is another way of another measurement that we use to figure out the oxygen carrying capacity of a person. So if you get your blood drawn, they may do your hematocrit, which we talked about that earlier. That's the number of red blood cells. So what percent of your blood is red blood cells versus plasma and white blood cells. You can also do a hemoglobin measurement. And hemoglobin, that's the actual compound inside the cell that binds the oxygen. And so in males, that hemoglobin should be between 13 and 18 per 100 mLs, and females 12 to 16 grams per 100 mLs. And I'm not too worried about you remembering those numbers. Normal values is something you'll learn and start to memorize later on. But you're probably thinking, well, if I know the hematocrit, why do I need to know the hemoglobin? Or if I know the hemoglobin, why know the hematocrit? Well, they're telling you two different things. Hematocrit is how many red blood cells there are, and hemoglobin is how much of this molecule that carries oxygen you have. So you may have a lot of red blood cells. You may have a really high hematocrit, but say your hematocrit's, you've got maybe a hematocrit that's maybe 10 times or 10% higher than normal, but you're still tired and you're sluggish, and you would think, well, if I've got plenty of red blood cells, why am I tired and sluggish? Well, you may not have enough hemoglobin. Okay, so it may be, if you were to have, some tiredness and sluggishness, your doctor may do this test and realize your hematocrit's fine, but your hemoglobin's low. So we've got to figure out, maybe we need to get you some more iron in your diet. We've got to do something to increase that hemoglobin so that you can carry more oxygen. You've got plenty of cells, just not enough hemoglobin. So that's why you measure both, okay, because knowing one does not necessarily give you the full story of why maybe you're being tired and possibly anemic. And we'll talk about different anemias in another video. So what is hemoglobin? So remember when we talked about protein structure back in the first anatomy lectures or first anatomy course, globin are those round proteins. And so you remember proteins have primary structure, then you can twist them into secondary structure, and you can twist them again to tertiary structure. And you can take several of these tertiary structures and bind them together to get a globin. So a globin is 4 proteins. So this is 1, 2, 3, 4. So you have 2 alpha chains and 2 beta chains. So you have 4 chains total. And each one of these chains carries this iron containing heme pigment. Okay? So the whole thing's hemoglobin, but in each one of these proteins you have this subunit called the heme pigment, and in that heme you have the iron. And so the iron is what really binds the oxygen. So if you think about taking a nail and putting it outside and it oxidizes, it's exposed to oxygen, it rusts, turn that red color. That's really kind of what blood's doing. When you have arterial blood that's fully oxygenated, it's really bright red. It's due to that oxygen-iron interaction. So each hemoglobin molecule can transport 4 oxygens. So you have 1, 2, 3, 4 irons, and each one of those irons can transport an oxygen. But each red blood cell has 250 million hemoglobin molecules. So you have 20 to 30 trillion red blood cells circulating in your body. So that's millions of hemoglobins, millions of red blood cells in each drop of blood. Tremendous ability to transport oxygen through your system. So hemoglobin, it picks up oxygen, and then it deloads or releases oxygen, and it also can pick up carbon dioxide. So we pick up oxygen in the lungs, okay? So when oxygen binds that hemoglobin, we call it oxyhemoglobin. Again, that's what gives arterial blood that ruby red look, that really bright red color of arterial blood is that binding of oxygen and hemoglobin. Once you carry that oxygen to the capillary beds in the tissues, you're going to download, you're going to take that oxygen and send it off into the tissues. It'll leave the blood, go to the tissues. Well, the blood will change color and become kind of a dark red color, that deoxygenated blood. And that's called deoxyhemoglobin, when you offload the oxygen. So oxyhemoglobin has the oxygen, deoxyhemoglobin, no oxygen. In addition, remember CO2 is a byproduct, a waste product of your cells. And so it's going to return to the blood vessel so that you can then take that to the lung, and that's what you blow off. You blow off CO2. CO2 is carried dissolved in the blood, but also part of it, about 20%, binds to the hemoglobin, and we call that carbaminohemoglobin. That was a hard one to say. I have to think about it. So some of the carbon dioxide that's carried through your blood is bound to that hemoglobin, and that again gives you a little bit darker blue color to the blood. All right, so in order to have enough red blood cells, we have to think about why we would make more or less red blood cells. So we're going to talk about